Hello students. So our next topic is oogenesis, which is formation of ovum in females. So here we'll see the process which happens in human. So in case of oogenesis in humans, it is said to be a discontinuous process. Why is it a discontinuous process? Because in females, when the female fetus is inside the mother's womb, then half of the process of this oogenesis is completed. And the later part, the maturation part, occurs after puberty. So since it gets arrested, the stage gets arrested in middle, means the process is divided before birth and after birth. That is why in humans, oogenesis or the formation of ovum is said to be a discontinuous process. So what happens is, we know what is gestation period. As the female fetus is growing inside the mother's womb, during the first month of gestation, about 1700. So we have already seen those follicular structure around the ovary. Those are the primary follicles. So about in the first month of gestation, about 1700 primary follicles are there in ovary. Okay, about 1700 primary follicles are formed. And in second month of gestation, those increase to about 6 lakhs because these follicles are continuously dividing. So 6 lakhs. In the fifth month of gestation, these 6 lakhs increase to about 7 million primary follicles. Now what happens to these 7 million primary follicles? These 7 million follicles will undergo, some of them will undergo atresia. Now what is atresia? Atresia is the shrinking of the primary follicles, means they degenerate, shrinking occurs. Okay, so after they undergo atresia, about 5 million will undergo atresia. So finally we'll have about 2 million primary follicles. Will have about 2 million primary follicles. So at birth, the ovaries will have about 2 million primary follicles and the rest will have undergone atresia. So this process continues till puberty. So in the gestation period, there are about 2 million primary follicles. So at puberty, there will be around 60,000 to 80,000 primary follicles. Okay, so in every 500 primary follicles, only one will grow to egg. So we won't find about 60,000 to 80,000 primary follicles. So it's 60,000 to 80,000 eggs. We won't find 80,000 eggs. In every 500 primary follicles, we'll get only one egg. Okay, so this occurs at puberty. From birth, 1700, then it will increase as the month of gestation goes on. 6 lakhs, 7 million, out of that 5 million will undergo atresia. So only 2 million will be left. Out of that 2 million at puberty, only 60,000 to 80,000 primary follicles will be there. Out of those, in every 500 primary follicles, only one will grow into egg. So now we'll see the different phases of oogenesis. Oogenesis is formation of ovum. So these primary follicles that are formed will start the process from these. Okay, so at puberty, what will happen? Okay, the whole process of oogenesis. Here is before puberty, after puberty, both included. Okay, so what will happen? The primary follicles will first undergo mitosis. Mitosis is equational division. That means the follicles will grow. The follicles will grow to form one oogonia. To form some oogonia which are diploid, twice in. Okay. Those oogonia, this is multiplication phase because multiplication is occurring, rapid division is occurring. So these primary follicles form oogonia, this is multiplication phase. It's same like spermatogenesis, 3 phases: multiplication, growth, maturation. So multiplication phases up to oogonia. Then these oogonia which are diploid will form the primary oocyte, which is again diploid. Means these oogonia will grow to form one primary oocyte, big one. So this is again diploid. So this is the growth phase. Growth phase, why? Because your oogonia has grown to form the primary oocyte. So here, what do we mean when we say it is a growth phase? Growth means here accumulation of yolk occurs, which is known as vitellogenesis. Okay, so yolk formation occurs, that is why growth occurs here. This growth is yolk formation. In humans, there will be very less amount of yolk, but yolk formation occurs, so this oogonia grows to primary oocyte. After primary oocyte, the maturation phase will come. Now, this primary oocyte will undergo meiosis 1, which is reductional division. Meiosis 1 to form 2 haploid cells, daughter cells. So, one haploid cell, see, one haploid cell which is smaller is the first polar body. This is not important, but this from this primary oocyte, one secondary oocyte forms. This is important. Here reductional division occurs, 2 cells will be formed, but this polar body is not required by us. We need the secondary oocyte to form the ova. So this secondary oocyte takes maximum of the cytoplasm part and this polar body becomes smaller, because this will degenerate later. This is not required. So this secondary oocyte is haploid. The secondary oocyte will leave the polar body. The secondary oocyte will again undergo meiosis 2. Meiosis 2 is again equational division like mitosis. So this will again form 2 haploid daughter cells. One is the second polar body, which is smaller again. Another one has the maximum cytoplasm part, which is our ovum or egg, or you can also call it ootid. Okay, so this is the process of oogenesis. Very simple. These primary follicles are formed. We start the cycle from this primary follicles. Primary follicles undergo mitosis to form oogonia. Oogonia undergoes growth to form primary oocyte. Primary oocyte will undergo meiosis 1 to form the first polar body and secondary oocyte. Secondary oocyte will again undergo meiosis 2 to form the second polar body and the ova. So this ova or ootid is that which gets released. The secondary oocyte is the one which is released during the stage. Means we have seen the picture in the structure inside the ovary before, where the Graafian follicle is formed. Then the secondary oocyte is released from there. So that stage, the secondary oocyte, here egg is released. From the secondary oocyte, egg is released. So ultimately, the ova will be formed. So this is oogenesis. One thing you have to remember is, when does oogenesis get arrested? Means when does it takes a pause. So oogenesis will get arrested in diplotin stage of prophase 1 of meiosis 1. This is when this oogenesis will get arrested. The process will come into a pause. So this is oogenesis. After this, we have to see this structure, the ovum structure in detail, the structure of the human egg. The egg has various membranes surrounding it, which is important because the sperm has to enter the egg. So structure of human egg. So the size is about 0.1 millimeter. 0.1 millimeter in diameter, and it is round. So now we'll see the different layers. There will be an outer layer, which is the plasma membrane. It is also known as the vitelline membrane. Plasma membrane or vitelline membrane. The next membrane, this plasma membrane is secreted by the cells itself. Okay, by the cells of the egg itself. Next membrane is the zona pellucida. This layer is secreted by the follicular cells. Zona pellucida. Then this is a cell. So there will be a nucleus. The nucleus here is not located in the center. It is located on one side. Okay. So this is the nucleus, or it is also known as germinal vesicle. Germinal vesicle, why? We'll come to know. This nucleus is located on one side. So the side where the nucleus is located is known as the animal pole. Okay, animal pole, nucleus is located, and through the animal pole only the sperm enters. The sperm will enter through the animal pole only. Okay, the other side, which is far away from the nucleus, this is the vegetal pole. Vegetal pole gives polarity to the egg. Okay, next, this part, outer part, which is dense, the dense outer part is known as egg cortex or exoplasm. This is dense. And the inner part, this is less dense. This is known as ooplasm. Okay, outer part is exoplasm, inner part is ooplasm. So what will the ooplasm consist of? The ooplasm consists of this nucleus and organelles. Along with the nucleus and organelles, it will also consist of the yolk, which is very, very less in case of humans. The yolk part is very less or negligible in case of humans. Okay, so these are the layers. Next, plasma membrane, zona pellucida. There will be another layer which are radiating like this. Throughout the egg there will be this layer. This is known as corona radiata. This is radiating outwards, so this name is corona radiata. These are also made of follicular cells. Made of follicular cells. So this is the structure of human egg. Okay, so this is done. Next part is you have to know the different types of egg membranes. One more thing you should remember, in humans, the egg, there is a name of eggs in different organisms. In humans, the yolk part is very less or negligible. So in humans, the egg is microlecithal. Microlecithal means very less. So where it has less yolk, so it is microlecithal. Now the types of egg membranes. Types of egg membranes. The first type is primary membrane. You just have to know the definition. One example. Primary membrane are secreted by cells itself. Secreted by cells. So here, what will be the example of primary membrane? this vitelline membrane. This is secreted by the cell. So vitelline membrane. Secondary membrane. These are secreted by follicular cell or made up of follicular cell. It can be secreted or made up of follicular cells. So if secreted by follicular cell, then the example will be zona pellucida. If made up of follicular cell, example will be corona radiata. So your example will be zona pellucida or corona radiata also. Next is tertiary membrane. Tertiary membrane is secreted by glands. Secreted by certain glands, especially in oviduct. Your example will be shell. Shell is also a membrane in certain organisms. It is present and these are secreted by glands. So it is tertiary. Okay. So in oogenesis, this you have understood the process. Next is the structure of human egg. This is something important. And next is types of egg membranes. So after this, the next part is we'll start with the in case of females, the cycle which is the menstrual cycle. So go through this part. After this, we'll start the menstrual cycle. Okay.